Antibodies, or immunoglobulins, can be subdivided into different classes according to their structure. There are 5 classes, called IgG, IgE, IgD, IgA, and IgM, each having different functions. The class of an antibody is determined by its heavy chains, and each antibody can be produced either as a cell surface antigen receptor on B cells or as a secreted molecule. The secreted form of IgA is a dimer of the basic 4 chain structure, and IgM is a pentamer. Naive B cells which have not encountered their antigen initially express IgM as their surface receptor. The heavy chain of IgM is called a mu chain, and a B cell makes a mu chain by transcribing its recombined VDJ gene together with exons for the constant domains of the IgM heavy chain. These are called C genes. C genes for the other immunoglobulin classes lie downstream of those for the IgM. An individual B cell can switch the class of antibody it produces. This process occurs in the germinal centers of the lymph nodes, spleen, and other secondary lymphoid tissues. Class switching is accompanied by a second process, somatic mutation, which increases the overall affinity of the antibodies produced. A primed B cell enters a germinal center and undergoes rapid division. At the same time, a mechanism is activated which introduces mutations into the DNA around the recombined VDJ gene, so each B cell has a slightly different antibody. Occasionally, a mutation will generate antibody with a higher affinity for its antigen. Follicular dendritic cells in the germinal center hold stores of antigen on their surface. B cells compete with each other for the antigen, and only B cells whose mutations have produced higher affinity antibodies will compete effectively for the limiting amount of antigen. This is now processed and presented to T cells, which signal division and differentiation, but B cells with low affinity antibodies have no antigen to present to the T cells and die by apoptosis. The interaction with T cells also promotes immunoglobulin class switching. Look now at the heavy chain gene locus. The IgM-C gene is preceded by a switching region which corresponds to switching regions preceding the C genes for all the other immunoglobulin classes except IgD. Here we see the event which occurs as a B cell switches from making IgM to IgG1. The C gene for IgG1 replaces the C gene for IgM, placing it next to the VDJ gene. Now the transcript from this encodes an IgG1 heavy chain, but with the same specificity as the IgM, since it uses the same VDJ gene which encodes the variable, antigen binding, part of the antibody. Exactly which switch will take place depends on the T cells and the cytokines they release, so the T cells determine what type of antibody response will be produced. B cells leaving the germinal center may become antibody-secreting plasma cells or memory cells. As a consequence of these processes, the affinity of the antibodies tends to increase during an immune response, and this is associated with a switch to production of IgG, IgA, and IgE. Secreted antibodies have many functions. The IgG antibody can act as an adapter. The 2 Fab arms of antibody have identical antigen binding sites. These form multiple non-covalent bonds with a specific antigen. The Fc portion of the antibody can link to Fc receptors on cells, including phagocytes and natural killer cells. This allows these cells to recognize their targets. For example, IgG can opsonize bacteria for phagocytosis by neutrophils. It can also activate the complement system, causing deposition of C3b. IgG is also transferred across the placenta from the mother to protect a child in the uterus and during its first few months of life. Meanwhile, other B cells may have switched to the production of IgE. This type of antibody binds to receptors on mast cells and basophils so that if they should contact antigen, they release inflammatory mediators. Other B cells, particularly in the Peyer's patches, may have switched to the production of IgA. IgA is produced as a dimer of the basic 4-chain immunoglobulin unit, linked by a joining or J chain. Cells of mucosal epithelia can bind IgA and transport it across to protect the mucosal surface. Some antigens, particularly polymeric carbohydrates, are not recognized by T cells and are called T-independent antigens. So the antibody response to these antigens shows neither increasing affinity nor class switching. The antibody against such antigens is predominantly secreted IgM, which is also the principal antibody in the earliest stages of an immune response to conventional T-dependent antigens. Because secreted IgM is a pentamer of the basic 4-chain structure, it is effective at cross-linking antigens and also fixes complement.
The main antigen-presenting cells are the B cell, the dendritic cell, and the macrophage. These cells can present antigen to CD4 positive cells. Additionally, all nucleated cells can present to CD8 positive T cells. All cells of the body express class I molecules encoded within the major histocompatibility complex, or MHC. Additionally, the B cell, the dendritic cell, and the macrophage express MHC class II molecules. These membrane molecules may contain degraded antigen peptides in a cleft on the top of the molecule, which they express to the T cells. The molecules regulate the way that the T cell reacts, since a T cell will only recognize foreign antigens when they are associated with the MHC molecules themselves. There are many varieties of class I and class II molecules, although in any one individual there may be up to 6 of each type. The MHC gene locus contains 3 class II genes, DP, DQ, and DR, each formed by 2 chains, beta and alpha. It also contains 3 class I genes, B, C, and A. Because each individual has 2 chromosomes carrying these genes, the maternal chromosome and the paternal chromosome, there are usually 6 different versions of the class I molecule and 6 different versions of the class II molecule, produced from the 3 different genes with the 2 chromosomes encoding them. Each of the class II molecules is formed from an alpha and a beta chain. The indent formed on the top of the molecule is where the antigenic peptide is held. Because the MHC is dependent on the genetic makeup of the individual, it is very unlikely that 2 unrelated people will have the same MHC molecules. Each of the class I genes produces a single alpha chain which folds itself up and interacts with another molecule called beta-2 microglobulin, which comes from a different gene locus. Since each individual has different MHC molecules, the performance of everyone's immune system is different. This affects susceptibility to diseases where the immune system is involved.
Immature lymphocytes formed in the bone marrow are released into the circulatory system. Some of these, which are to become T cells, must first undergo selection based on specificity and degree of response to antigens. This occurs within the thymus gland located in the cardiothoracic cavity above the heart. Within the thymus, pre-T cells migrate to the subcapsular region. These cells then divide rapidly and pass into the cortex. At this time, each will produce its particular diversified T cell receptor. This pre-T cell must also generate 2 other receptors, CD8 and CD4, which will enable it to bind to the major histocompatibility complex molecules which present antigenic peptides. All these cells are termed thymocytes. They now migrate deeper into the cortex and pass among large numbers of epithelial cells which have MHC class I and II molecules on their surface. It is the CD8 receptor, together with the T cell receptor, which may bind to the MHC class I molecule. Alternatively, binding could occur through the CD4 receptor with MHC class II molecules. Cells which fail to bind to either MHC molecule type die by apoptosis because they are not given the signal necessary to rescue them. A large proportion of thymocytes are lost at this stage, which is termed positive selection. Surviving cells continue towards the medulla. Interdigitating cells present them with antigens belonging to the body. Those which bind too strongly are destroyed. This is called negative selection. The cells which do survive, possibly less than 1% of the original population, now lose their non-functional MHC receptor, CD4 or CD8. Those which respond to MHC class I keep the CD8 molecule, but lose CD4. Conversely, thymocytes which bind with MHC class II through CD4 lose their CD8 group. They are now mature T cells. The educated T cells pass through the medulla back into the circulation. The bloodstream carries the T cells to the secondary lymphatic tissues, where they remain until stimulated by antigen, presented on antigen-presenting cells.
The body is capable of two major types of immune response to an infection: the production of antibodies, illustrated here on the left, and cell-mediated response on the right. With antibody production, a B cell presents antigen to a Th2 cell. This triggers the release of cytokines including interleukin-4, interleukin-10, and interleukin-13. The B cell then divides and differentiates into plasma cells producing antibody. With cell-mediated response, a macrophage internalizes material and interacts with a Th1 cell, which causes the production of cytokines, including interferon gamma, by the Th1 cell. This is capable of activating the macrophage and inducing MHC class II molecules on its surface. When an antibody response takes place, interleukin-10, produced by the Th2 cell, will prevent Th1 cells from producing interferon gamma, effectively suppressing the cell-mediated response. If a cell-mediated response occurs, the interferon gamma, which activates the macrophage, also inhibits proliferation of the Th2 cell, thus suppressing the antibody response. IFN gamma also acts on B cells to modulate antibody class switching. The immune system therefore goes into one mode of response or the other. Another layer of activity which affects the type of response which predominates is the production of interleukin-13 cytokines by the Th2 cell in an antibody response. This blocks the production of interleukin-1 and tumor necrosis factor, which are the prime effector cytokines of the macrophages. It also promotes B cell proliferation. With the cell-mediated response, the production of interleukin-12 by macrophages tends to promote differentiation of T cells into its pathway, loading up more cells which can reinforce this aspect of the response. Here we see a blood vessel lined with endothelial cells with a normal flow of blood containing lymphocytes. We are going to follow one particular lymphocyte to see how it migrates through the endothelium and out into the surrounding tissues. As the vessel widens into what is known as a venule, the blood flow decreases and the lymphocytes slow down. In the presence of an infection, such as bacteria, an immune response takes place, and cytokines, such as interleukin-1 and tumor necrosis factor, are released from tissue cells and resident macrophages. In addition, peptides of the complement system cause mast cells to degranulate. Cytokines, such as interleukin-1 and tumor necrosis factor, act on the endothelium to induce adhesion molecules, while chemokines attach to the endothelium to act as stimulators of migration. Cytokines induce the expression of adhesion molecules on the surface of endothelial cells complementary to those on the lymphocytes. Molecules on the endothelial cells, such as E-selectin, interact with carbohydrate molecules on the surface of a lymphocyte, such as CD15. These interactions cause the lymphocytes to slow down and roll along the endothelium, enabling them to sample other adhesion molecules which have been induced onto its surface. Amongst these are the chemokines, which bind to the lymphocyte and trigger an activation mechanism, causing an influx of calcium ions into the cell and the activation of another group of adhesion molecules on its surface. These adhesion molecule proteins, called LFA1 and VLA4, interact with other adhesion molecule proteins on the surface of endothelium, called ICAM1 and VCAM1, respectively. These latter molecules can also be induced on the endothelial surface by cytokines such as tumor necrosis factor, thereby enhancing binding just at this site. This multi-point adhesion onto the endothelium is called pavementing. Both the adhesion molecules on the lymphocyte and those on the endothelium are each attached to the respective cell's cytoskeleton. This can be thought of as a molecular motor which acts like a tractor, enabling the lymphocyte to pull itself down and out through the endothelium. With a firm anchoring point to move against, the lymphocyte proceeds to extend its pseudopod down into the intercellular junction between the endothelial cells, which retract as they are pushed aside. When the pseudopod makes contact with the basement membrane, the cell moves new adhesion molecules down to its leading edge, enabling it to form a new anchoring point. The lymphocyte now releases granules from this leading edge. These contain destructive enzymes such as collagenase and elastase, which puncture the basement membrane, allowing the cell to squeeze down through the damaged area and migrate out into the tissue. This migratory process is called diapedesis. The damaged endothelium then seals over again and reforms. The lymphocyte then migrates toward the site of infection under chemical stimuli in a process known as chemotaxis. Another feature of inflammation is an increase in capillary permeability. Mediators from activated mast cells, such as histamine, cause the endothelial cells to retract. This allows larger serum molecules, such as antibody and complement, to leak into the inflamed area. These mediators can also act on the smooth muscle in the blood vessel wall, increasing the diameter of arterioles and producing enhanced blood flow. This causes increased supply of serum molecules and cells to the inflammatory site.
Here we have a population of mature B cells expressing surface antibody and educated T cells with T cell antigen receptors on their surfaces. At any one time in a population of lymphocytes, there will be millions of different clones of cells, each carrying a different receptor, and therefore only able to recognize a particular type of antigen. This means that in a normal immune system, there is always a small subpopulation of cells which are able to recognize a specific antigen. If this were not the case, we would be completely unable to protect ourselves from pathogens. Such cells are said to share the same specificity. This is one of the reasons why lymphocytes circulate around the body, to ensure the small number of cells which are able to recognize a specific antigen will actually come into contact with it. Consequently, most of the cells in the human body are never called upon to fight infection. They are simply there just in case they are needed to combat any new strains of pathogen that might enter the host. We are going to look at how B cells and T cells differentiate and the mechanisms which bring this about. Here we have a virgin lymphocyte pool. In the presence of an antigen, only one of these many different B cells is able to bind to that antigen successfully. This process, known as clonal selection, initiates a primary immune response which causes the cell to undergo a series of irreversible changes. The antigen is taken up from solution by the B cell's surface immunoglobulin. It is internalized and processed. Antigen fragments are subsequently re-expressed on the B cell surface associated with MHC class II molecules for presentation to Th2 cells. Receptors for interleukin-4 are also induced on the B cell. Presentation of the antigen to the Th2 cell causes vesicles within the T cell to fuse with the cell surface and release cytokines, including interleukin-4, which then binds to the interleukin-4 receptors on the B cells. Together with costimulatory signals, the IL-4 activates the B cell, triggering its division. Other cytokines released from the Th2 cell, including interleukin-2, interleukin-4, interleukin-5, and interleukin-6, then bind to the appropriate receptors on the B cells, causing them to differentiate. Some of the cells become plasma cells which produce a secreted form of the original antibody. Consequently, they are also known as AFCs, or antibody-forming cells. Plasma cells are a different shape from B cells and have lost all their original cell surface antibody. They will go on dividing several times, but within days or weeks they will die. Other cells become memory cells. They have undergone a series of phenotypic changes, making them more efficient at reacting to the same antigen, if it is ever presented again in the future. This means that there is always a ready supply of cells with the appropriate specificity to fight infection. These cells can live for many years, and this increased efficiency of the memory cells to fight infection underlies the enhanced secondary immune response. Memory cells confer lasting immunity against a pathogen, which is the basic principle behind vaccination. Now we shall look at how T cells differentiate. The process is quite similar. Again, we have a pool of virgin lymphocytes. There are millions of different clones of T cells, each carrying a different T cell antigen receptor. In the presence of an antigen-presenting cell, such as a macrophage, only one of these many different T cells is able to bind to that antigen successfully. As we have seen, this is called clonal selection. The antigen-presenting cell interacts with the T cell and releases cytokines such as interleukin-1, which cause the expression of interleukin-2 receptors on the surface of the T cell. There are also crucial interactions between pairs of co-stimulatory molecules on the surface of each of the two cells. In addition to being able to bind interleukin-2 using its new receptor, the T cell is now also able to produce its own interleukin-2, enabling it to stimulate itself. This ability to produce cytokines, which can act on cells of the type that produce them, is called an autocrine action, whereas producing cytokines, which stimulate another cell type, is a paracrine action. The interleukin-2 binds to the receptors on the surface of the T cell, triggering division. This causes the induction of new co-stimulatory molecules on the surface of the T cell. The T cell divides, and after 5 or 6 cycles there is a gradual loss of the interleukin-2 receptor. This prevents the cells from receiving any positive signals to continue dividing. All of the T cells eventually lose their interleukin-2 receptors but retain their T cell antigen receptor. However, some of them also retain the signaling co-stimulatory molecules, and those become the memory T cells. As before, these cells are able to mount an effective attack against the specific pathogen in the event of future infection. Memory cells remain in the lymphatic system where they can live for many years. Other cells lose their co-stimulatory molecules and become effector cells, which migrate out into the surrounding tissue to participate in effector responses to eliminate infection. These cells die within a few weeks. Both B-cell and T-cell clonal proliferation is driven by the presence of antigen. As soon as the antigen has been destroyed, the whole immune response switches off, leaving more cells in the system sharing the same specificity than existed prior to the original infection. In the presence of infection, chemotactic molecules are released from the complement system, mast cells and leukocytes. These in turn stimulate macrophages to bind to endothelial surfaces in blood vessels near the site of infection, facilitating their passage through the endothelial wall. Once the macrophage is within the tissue fluid, it migrates towards the site of infection across the surface of the extracellular matrix. This migratory process is termed chemotaxis. Its direction is controlled by the concentration gradient of free chemotactic molecules, such as C5a. The macrophage achieves this migration by means of surface integrin molecules, which are able to bind to proteins within the extracellular layer as long as the cell moves up a chemotactic gradient. Each molecule of integrin attaches to the extracellular surface, then is moved by the cytoskeleton towards the rear of the macrophage. On reaching the trailing end of the macrophage, it detaches and is reabsorbed within the cell. Other integrin molecules emerge from the leading surface to continue the process. Once the chemokine concentration ceases to increase, receptors are no longer stimulated and the integrin stops binding to the extracellular surface. The macrophage, therefore, comes to rest as it is now at the intended destination. The macrophage is equipped with CR3 receptors for the C3b residues deposited on the surface of the target bacterium by the complement system. When this binding takes place, the antimicrobial mechanism is triggered. The bacterium is held and enveloped by the macrophage and is endocytosed, a process called phagocytosis. The macrophage attacks the enclosed bacterium in a variety of ways. Enzymes assemble in the phagosome membrane, which can reduce oxygen by the addition of an electron to form superoxide anions. These anions further react to form a variety of reactive oxygen intermediates which can directly damage the bacterial membrane. Within the body of the macrophage, there are small vesicles called lysosomes containing enzymes. When a bacterium is endocytosed, the lysosomes may fuse with the vacuole and release an enzyme called myeloperoxidase. This can act further on the peroxides to form even more chemically reactive species. These processes constitute the oxygen-dependent mechanism of bacterial killing. In the first 15 minutes after the fusion of a lysosome with the vacuole, the pH rises. During this period, cationic peptides called defensins are able to penetrate bacterial membranes and create channels for ions to enter the bacterium, disrupting its metabolism. Additionally, an iron chelating agent, lactoferrin, is released. This binds free iron, denying it to the bacteria which need it to reproduce. Subsequently, pH falls as hydrogen ions are pumped into the phagosome. This tends to inactivate the defensins but creates a suitable environment for other lysosomal enzymes which attack bacterial membranes. The lactoferrin is also active under acidic conditions to inhibit bacterial division. When destruction of the engulfed organism is complete, debris is ejected. The preceding process will occur even if the immune system does not specifically recognize the bacterium. If, however, Th1 cells exist with receptors for the bacterial proteins, they recognize bacterial products presented by the macrophage and release cytokines, notably interferon gamma. These cytokines stimulate the macrophage's killing mechanism, increasing its ability to destroy bacteria with oxygen radicals. Also, another pathway which is not oxygen dependent is activated. This pathway generates reactive nitrogen intermediates, such as nitric oxide, which attacks bacterial membranes. The other mechanisms seen in non-activated macrophages are still found but occur more intensely. Lysosomes release cationic proteins which may penetrate bacterial coatings during the high pH alkaline phase, followed by lytic enzymes which are active in the acid environment which follows. The activated macrophage destroys engulfed bacteria rapidly, and through the use of reactive oxygen and nitrogen intermediates will deal with some varieties of bacteria not susceptible to attack by non-activated cells.